Okay. Hello, everyone. I'm Lakshmi Subramanian, Program Manager of the Seven Bridges Cancer Genomics Cloud. On behalf of Seven Bridges and the Cancer Genomics Cloud resource teams, I would like to thank each one of you for joining us today. So Seven Bridges is a global biomedical data company which provides end-to-end informatics infrastructure for several disease-focused research initiatives. Today, you're going to learn about one of the three cloud resources funded by the National Cancer Institute, and it's called the Cancer Genomics Cloud. CGC allows cancer researchers to explore and analyze massive public data sets like the Cancer Genomics Atlas or TCGA, or the Clinical Proteomic Tumor Analysis Consortium, that's CPTAC, and several other large multi-omic data sets on a secure and scalable computational platform. We are also involved in other consortia, such as the Gabriella Miller Kids First, or the Pancreatic Cancer Action Network, or PanCAN, and the NHLBI Biodata Catalyst. So today's webinar is the inaugural webinar of the CGC monthly webinar series. So although this webinar is aimed to provide more introductory information about the CGC, in the upcoming months, we'll have a variety of interesting topics, such as how you can use the common workflow language and so on. So I just want to introduce the CGC monthly webinar series to all of you. So the goal is for everybody to learn about the CGC platform features that you can use in your own research projects. And like I said, the topics are going to be a variety of research as well as the technical topics in the field of cancer research, particularly using the Cancer Genomics Cloud. And all the resources, including this slide deck and the recording of this webinar, and information and announcements about upcoming webinar topics and speakers, slides and everything will be made available at this website. So all you have to do is maybe bookmark this page so you can get back to that for accessing the materials. And these webinars are going to happen every month, 4th Wednesday at the same time, 2 p.m. Eastern. So please feel free to save the date and time, and you will receive the link to register. And if you have any questions, because of the size of the webinar, we have muted everyone except the speakers. So if you have any questions, please feel free to use the chat box for asking your questions. And at the end of the presentation, we will get to as many questions as possible. And we will also send a link for a survey. So we request you to kindly provide your feedback. So let me go ahead with my with extending my acknowledgements to the entire Global Seven Bridges team for making the CGC possible, and also the CGC users and collaborators. I would like to thank our funding agency, the National Cancer Institute. CGC is part of the Cancer Research Data Commons. So I would also like to thank our other cloud resources at ISB, Institute for Systems Biology, and the Broad Institute, and the Data Commons framework services provided by the University of Chicago. All right, so let's get started. I want to start off with a research problem, okay? So just like all of you are working on some different area of cancer, let's consider this research user who is working on pancreatic adenocarcinoma cases, okay? And the user wants to identify the mutational burden. That is, what are the top mutated genes and the somatic signature in pancreatic adenocarcinoma cases? As you can see, there are like lots of varieties of data sets available across the board. And the researcher has several questions and concerns. Some of them I have listed here. Like, for example, you may think that I need access to all these different data sets. I need to see them in one place. But that's not enough, like if I have the data sets, but I don't have access to the tools, then what do? So I need access to the data sets, tools, and the workflows all in the same place. That's one question. And then research doesn't happen in a silo, right? Like you want to share with your collaborators, with other people in your own lab or outside your laboratory across the country or the world. So you need a way to securely share all the results of your analysis with your collaborators. And then there are lots of public data sets available, but then you are doing your own sequencing experiments and you want to analyze them safely and in a secure way in the cloud. So what do? There's another question that comes to a lot of people's mind. These days, all the sequencing machines dump all the data, the raw files, on some cloud. It could be on Amazon cloud. It could be on Google Cloud. So you're worried that your data is just available in a certain cloud, and you want to use a cloud resource which is set up on a different cloud. So that's a challenge. And then consider the situation where, let's say you're a postdoc in the lab, you're performing analysis on a set of tools and workflows, and the PI decides that you want to redo the same thing on using a different set of parameters, or you come up with a different strategy. You want to redo the analysis using a different set of parameters. So you don't want to start from scratch. You want to be able to pick up from a certain point of your workflow and then rerun the analysis. And finally, this is the biggest concern several people I've spoken with. So you're worried about cloud costs. It's a general thinking that doing anything on the cloud is going to be super expensive, and you're worried about how can I handle billing? How do I bill this to my grant? A lot of questions, and I hear you. So my elevator pitch today is, in this next 1 hour, we want to alleviate all these concerns for you and then show you how you can use the Cancer Genomics Cloud for successfully doing your research analysis. So with that kind of introduction, I would like to just review the agenda for today's presentation. Dave, my co-presenter, I would like to introduce Dave, who is the Community Engagement Specialist at 7 Bridges.
Hello.
Hi, Dave. Okay, so Dave is going to walk us through the background of the CGC, and then I will review our collaboration and engagement efforts, and also introduce you to the different multi-omic data sets. And then Dave will again review the CGC features and also show you a demonstration of an example analysis on the Cancer Genomics Cloud. And we'll wrap up with questions and discussion. Over to you, Dave.
Thank you, Sai, for that great introduction. So I'm excited to be here. Let's start off with background on the Cancer Genomics Cloud and how everything got started. So many of you have seen a slide similar to this, but there really is an explosion of genomics data, which has been spurred on by the ease of sequencing and lower cost per base of sequencing. And so this has resulted in the cumulative number of human genomes and exomes exponentially increasing over time. And it has also enabled population scale sequencing efforts, the most well-known of which is the Cancer Genome Atlas created by the National Cancer Institute. And ultimately, this got started maybe 14 years ago or so. And ultimately, this involved 33 different tumor types and a lot of rare cancers, actually, approximately 11,000 patients. The total of this data is over 2.5 petabytes of data. And it's a very powerful and unique data set, but presents some challenges. One, how do you get secure access to the files you want to analyze? And two, how can we analyze the data efficiently and not move the data around unnecessarily? So that was the impetus for the cloud pilots, which eventually grew up and matured into the National Cancer Institute's Cancer Research Data Commons. So this is a new paradigm of doing cancer research. And as you can see, all the data nodes that we're going to talk about today are in the middle. So the TCGA is actually just one section of the genomic data commons, but there are other data nodes such as the proteomics data commons and others, which Sai will get into in the next section. And so this paradigm is that the data contributors and consumers log on to one location and they bring their expertise, they bring their software and their data science skills to do the analysis in one location. So you no longer have to move the data around unnecessarily. But on top of that, to add value, we have authentication services. We have validated tools, elastic compute, and all these things that create a ecosystem for cancer research. And this is important because multi-omics is becoming increasingly critical for cancer research. So it's not just the somatic and germline mutations, but it's also the epigenome, the transcriptome, proteome, etc. And this will help us go from population studies into the era of precision medicine, where we can actually create new cancer treatments. And so you have to integrate more than one data set now to get a comprehensive picture and characterization of what's causing cancer and what could also be used to create a new type of treatment. Okay, and so things started out for the Cancer Genomics Cloud in 2014, and then over time, more data sets were added. As you can see, here's some of the logos on this timeline. Most recently, we've integrated with the ICDC, or the Integrated Canine Data Commons, as well as the Proteomics Data Commons and the Cancer Data Service. As of this month in 2021, Genomics Cloud. So researchers are getting a lot of use out of this and running analysis in parallel. And that's primarily because the CGC is democratizing complex analysis in a fair data ecosystem. So FAIR is an acronym that stands for findable, accessible, interoperable, and reusable data. So that theme is pervasive throughout the entire CRDC ecosystem. And some of the features I'm going to talk about later include easy data management, very important secure collaboration and managed billing, as well as scalable compute and optimized and flexible bioinformatics. So keep those things in mind when we talk about the important components of the CGC. But what we've seen is an acceleration in cancer research. So users are logging on and they're doing HLA typing and microRNA expression analysis. They're looking at viral infection across patient populations and doing a lot of RNA-seq analysis as well. And this is because the CGC provides an easy way to not only find, but also analyze your data. And we'll go through a demo of this today. So we can, there's multiple ways to find data. You can actually, what's that?
Just a second. I see there's a chat message. The user saying they cannot hear the speaker. So I'm wondering if others have the problem as well. Maybe you may want to check your connection or the mic.
Can you hear me, Sai?
I can hear you.
Okay.
I don't know if the other speakers can.
Okay.
Okay.
So let us know if anyone else has a problem.
Okay. I think they're saying that if they reconnected, it worked. So maybe that's something everybody can try. Okay. Sorry about that. I hope it works for everybody. Okay.
Oh, so a lot of people had to reconnect. Okay.
Exactly. I think we can go ahead.
Okay, great. So we'll go through an example using a TCGA analysis, and you can query things and do your analysis using API, doing commands either in RStudio or Jupyter Lab, but you can also use some of these interactive visual query systems that we have, such as the data browser. Okay, so how do you get started, everyone, on the CGC? So it's very easy to get started. The main website is cancergenomicscloud.org. And when you go to this website, you'll see the menu bar here. If you click login, it'll take you to a new screen. And there's two options for you. You can log in with your, you can create an account with a username and password combination, but you can also attach your ERA Commons ID. And that'll automatically not only create an account for you, but also forward any dbGaP permissions that you have, which will be helpful for accessing the controlled data. But that said, there's plenty of open TCGA data to get started, and that's the example we'll use today. The NCI has provided $300 in pilot funding. So that's anyone who makes an account gets that funding, and that can be used to get your project started, to test some of your pipelines you may want to run, and to get an idea for how much it'll cost when you scale up to do your full analysis. But there's other funding opportunities, which Sai will mention in the next part. And of course, we have technical support via documentation, and you can message the SB team at any time. Okay, so I'll pass it back to Sai, who will talk about collaboration and engagement.
Excellent. Thank you so much, Dave, for the very nice overview and to learn about the history of CGC. I think it was really exciting to hear that. So I'm going to talk about the collaboration and community engagement activities. So Dave introduced that we'll be getting $300 worth of cloud credits. So that's a good amount of money to process a bunch of whole exome sequences and also maybe do some RNA-seq and differential expression analysis to get a feel of the platform. But beyond that, you know, if you run out of that funds, but you have some really excellent ideas that you want to try out for your research, we have this opportunity called the Collaborative Project Program. So you can submit a proposal and you will be awarded up to 10,000 papers to different journals from just a single project. So we highly encourage applications from students and postdocs. So as a case study, I want to highlight this collaborative project that we did with Xavier, who is from the Center for Cancer Research at NCI. So Xavier used the CGC to understand microRNA biogenesis in cancer. So as you can see in this timeline, he submitted an application for collaborative project in 2017. And together with our team, Xavier developed this tool called Quagmire, which is used for pulling specific reads and aligning them in the middle of a microRNA. So the idea was initially to reprocess all the TCGA miRNAs with Quagmire. But the CGC provided such an efficient and scalable environment for his analysis. It led to several more research projects and a second collaborative application in 2019. And as you can see, 3 papers came out of this work. So this is a very good success story I wanted to highlight. And if you notice, Quagmire is now available for anybody in the CGC public apps gallery. So you can use it. And Xavier has also published a YouTube video. You can actually learn how to use Quagmire in less than 2 minutes. And you can also visit the GitHub page to get more details about this tool. So you can do these kinds of collaborative projects as well. So please feel free to reach out to us. So moving on, I want to highlight how the Cancer Genomics Cloud has been used as a platform for supporting data commons and a coordinating center. So 7 Bridges, together with the Jackson Labs, formed the PDXNet Data Commons. So we were awarded this grant with Jackson Labs, and PDXNet is a large consortia for development and preclinical testing of targeted therapeutics in patient-derived xenografts, which are mouse models, to advance cancer precision medicine. So you can see there are several centers across the country who are, working on patient-derived xenograft models, and all the data is stored and hosted in the CGC. And there's a portal which I've linked here. You can visit that to learn more about this. But if you stay tuned until the end of this seminar, actually, I want to tell you something very exciting and how you can learn more about the PDXNet. And I want to highlight that there have been several high-impact publications, not just from PDXNet, but from several of our collaborative projects. And I've just highlighted a few of them here. And these are from 2020, like some of the latest publications from our collaborative project users. And the one on the right, which is the Nature Genetics paper, was just published earlier in January. It's from the PDX group. And they have highlighted how PDXs are extremely efficient for preclinical testing. Okay, so moving on. So we saw how CGC can be used as for collaborative data analysis and also to support large research consortia. But if you notice that CGC is a platform which has been excellently set up for training needs. So NIH is making lots of investment on data analysis ecosystem and platforms. So we empower all our researchers, and we also want to empower students to learn from the Cancer Genomics Cloud platform. So this CGC can be used as an excellent teaching tool as well. So as a use case, I want to highlight this master's program in health informatics and data science at Georgetown University. So we were invited to teach a bioinformatics class for the master's students in both 2019 and also in 2020. So students use the CGC as a platform for learning how to set up public workflows or pipelines and use them in their own data analysis. And of course, the Georgetown faculty follow up with more detailed technical training for the remainder of the semester. And it has worked out excellently, as a teaching aid. So students learn, in the last 2 years, the master's students have learned how to create differential expression workflows and also to perform single cell analysis. So that's great. Okay, so I want to highlight how we are participating in several open standards groups, and this makes the CGC more interoperable and also enhances workflow portability as well as reproducibility. So we are active members of the Global Alliance for Genomics and Health, which is called GA4GH. So we have implemented several of their standards for workflow execution, task execution, and so on. We're also pioneers in developing the common workflow language, which makes it very easy for stitching together several pipelines and dedicated workflows for your research analysis. We actively deposit our tools in the doc store, so it's accessible by everybody. And also, we are actively involved in the biocompute object implementation, which is a standard. It's an IEEE standard designed for communication of next-gen sequencing data, and it's supported by the FDA for regulatory submissions. So I will hand it back. Oh, sorry. I'm going to be introducing multi-omics data sets in the CGC in my next section. Okay, so you can search and access several large public data sets on the CGC. So this is like a cheat sheet for you all to look at any time to see, okay, what kind of data is available in the CGC. So I see there's a question in the chat, are any of the publications using CRDC data? So a lot of the publications are using CRDC data. So as you can see, several of these data sets come from the Cancer Research Data Commons. So we have data from the TCGA, which is the largest data set, from over 11,000 patients covering 33 cancer types. These data sets are from a variety of experimental setups like whole exome, RNA-seq, 8-hack-seq, imaging, and so on. We also have data sets from childhood cancers, which come from this consortium called Target. And we have imaging data, which is mostly radiology images from the TCGA cohort. So from about 21 tumor types, we have imaging data, which is stored in a standard format called DICOM. And next is the CPTAC, which is the Clinical Proteomic Tumor Analysis Consortium. So CPTAC is interesting because it has, it's primarily for proteomics, so it has mass spectrometry data, but you also find associated genomic data for certain tumor types. So it's very interesting, and CPTAC is also available, both proteomic as well as the whole genome, exome, and RNA-seq data sets are available on the CGC. And going a little bit further, we have brought in the International Cancer Genome Consortium, which is the ICGC. Over 20 studies are available for you to access and explore. We have cancer cell lines from the Cancer Cell Line Encyclopedia, which is the CCLE. And there are a few other projects like the Simons Foundation, which has led to genomic sequencing of 130 different populations. So there's whole genome sequencing data from that. And then there's this PGP UK, which is the Personal Genome Project, which has data sets from several individuals. And finally, single cell genomic data set from healthy tissues, which are from the Human Cell Atlas. So as you can see there, these are a variety of file types. So you have access to raw sequencing files, which are the FASTQs, and then you have access to the alignments like BAM files, and then you also have variant calls in VCF formats, and there are data available in other formats like the DICOM images, text files, and so on. So there's so much variety available for you to explore and analyze. And CGC connects with several cancer research data commons, that's the CRDC data repositories. So I want to highlight that we have data from the genomic data commons. So TCGA, Target, and CPTAC come from the genomic data commons. And then, like I mentioned, CPTAC is the proteomic data, which is the mass spectrometry data from large consortia. And then we have the ICDC, which is the Integrated Canine Data Commons. So it's a cloud repository of canine cancers. So the goal is you should be able to do comparative analysis on human cancers with canine cancers. So that data is made available. And recently, we integrated with the Cancer Data Service, so which has several NCI-funded data sets, which don't fit into any of these existing data commons, but are still very valuable for researchers to access. So we made that available via the CGC. And CTDC, which is the Clinical Trial Data Commons and Imaging Data Commons, we will be bringing them soon this year. So I wanted to highlight another use case, which is our successful integration with the proteomic data commons. So you can see that this is a paper that was published on quantitative proteomic landscape of breast carcinoma subtypes. And also there was another paper which came, which is on clear cell renal cell carcinoma, CCRCC. So this group at University of Michigan, our collaborators, they used 3 different tools called MS-Fragger, Philosopher, and TMT Integrator. So they use this technology called Tandem Mass Tag, which is TMT. It's a proteomics technology, and they use the Cancer Genomics Cloud. Proteomics tools can run very quickly and efficiently in the cloud. So this lowers barriers for usage. You can try bringing your own tools, stitch a pipeline, and then take advantage of the full breadth of cloud resources that are available. So the time for analysis was just 4 hours, on 23 different AWS nodes. So this actually highlights, and the cost was just about less than $30 for the total analysis. Over 0.5 terabytes worth of proteomics data was analyzed. So this just highlight that you can benefit from using the cloud, and you can, save cost as well as time. Moving on, I'm going to hand it back to Dave, who will introduce you to the TCGA analysis, highlight the CGC features, and also show you a demo.
Thank you, Sai. So I'm going to start with the introduction to TCGA analysis. I just want to let everyone know that we're recording the webinar. So if you have audio issues, you can view the recording later. So we started off with a question, a research question of how do I identify the mutational burden, top mutated genes, and somatic signature in pancreatic adenocarcinoma cases. So hopefully, this researcher has some concerns. So hopefully we alleviated a couple, and I will address some of the other concerns as I talk about the important features of the Cancer Genomics Cloud. So the software I'm going to highlight right now is called MAF Tools, and it's a part of the Bioconductor project, and it was published a couple years ago. And I think this is a really great way to get started, a piece of software to get started to analyze the TCGA data. And the reason I say that is the MAF files for the somatic calls are open. So even if you do not have access to some of the controlled levels of TCGA, you can access all 33 studies, access the MAF files, and then use them in an analysis. And the package itself actually offers a variety of visualizations and analysis. So today, to answer our research question, we're going to use the OncoPlot visualization function. And we're also going to use some of the somatic signature functions as well. But this package will also analyze the open copy number variation data as well. That said, you do not need to do any R coding to do this demonstration because we're going to use some of the apps that are available. Okay, so after doing some training for a variety of users, I've identified that there's a typical user flow that any type of user will generally go through, no matter if they're doing map tools or they're doing RNA-seq, etc. And that involves 4 steps. So the first is to create a project and then find your data and link it back to your project. And then they move on to finding the tools that they need, and they organize those in their project. And then they finally run their analysis. And there's 2 ways to run analysis. You can actually run a task, which I'll talk about later, or you could interactively create notebooks in RStudio or JupyterLab, both of which are available to you. So everything starts with a project, and projects are very important. On other platforms, they're sometimes called workspaces or sandboxes, but you can think of them as a container for your files, which are your raw data. So in this case, we're going to actually use open math files from TCGA. So those will be our starting files. And then you can contain your app. You add your apps to the project. And then when you run an app and you have a specific analysis, that's called a task. The result of the task are new derived files that are added back to your files collection. And then you can iterate over the new files and do new apps like that. Projects are also how you can do collaborative science by adding new members to your project. And when you add new members to your project, you can fine-tune their permissions and give them either read-only access or execute, or they could be co-administrators to your project as well. And it's very easy to share results by adding new members to your project. But when you make a new project, there's actually a large amount of flexibility. And I want to review some of these things right now. When you make a new project, you can customize the URL, and you can actually select a billing group. And I'm going to talk more about this later. But just keep in mind, you have a pilot funding billing group with $300 in cloud credits. In addition, you have NIH Strides discount that's automatically built into any of the storage and the compute as well. So you don't have to bring in a new NIH Strides account. So you automatically get that discount as a NCI researcher. You can choose to use spot instances, which are reduced cost computation, and as well as some other features. Most importantly, I want to highlight that we actually operate on AWS and Google, which will come important later. There's another slide for that. But let's consider the billing group. So everyone will have a pilot funds billing group that you can use for testing. But if you decide to apply for collaborative funds, that would be a new billing group. And you can also belong to other billing groups for your collaborators. So the point is, you do not need to bring in a new Google account that's attached to a credit card or Amazon account, because that's already built into the Cancer Genomics Cloud. And that's very, very helpful because your analysis charges, the total charges are broken down into analysis charges as well as storage costs. And you can feel free to email the SB team if you're worried about your daily storage cost as well. Like I said before, we actually operate on a multi-cloud. So we have a core platform infrastructure that operates on one cloud, but then you can create projects that do the analysis and storage on either AWS US East 1 or Google US West 1. And you can make use of any of the compute instances in either platform, as well as the object bucket storage. And this makes the platform very flexible because a lot of times users are concerned, maybe they have a sequencing center that's delivering BAM files or VCF files on a Google bucket. They don't want to pick the wrong cloud. So now you can actually interchange across different clouds, and you're able to actually analyze the data where it resides, saving time and your funding as well. Memoization is a great feature that I want to talk about. So The platform actually can remember your intermediate files, not just your full output files, but your intermediate files, and you could save time when you recompute using different parameters, or when you add samples. So let's say you're scaling up from 10 to 100 subjects. The platform will remember what compute has already taken place and reuse those results as well. This is helpful for a complex pipeline such as whole genome sequencing, BWA plus GATK. So we talked about all the project options. So let's talk about how you may find and access data. So there are many options to bring in data to the platform. And here's a screenshot of the add files window. And we're going to talk about data browser and what that enables later. But we also have a lot of public files to get you started. So there's a lot of test files. There's also a lot of human genome references. Those are all hosted on the CGC, so you don't have to upload those or pay for storage for those. You can actually upload directly from your computer using the web browser, so that's helpful for smaller bed files or manifest files. You can link, like I said, Google buckets or Amazon buckets and have those appear as natively part of your project. We also have data tools such as a command line uploader. So if you're using the NIH BioWolf, you can upload directly from that using the command line uploader. There's also a desktop uploader that works on Windows, Mac, and Linux. And you can import via Manifest, which I'll talk about shortly. But today we're focused on pancreatic cancer. And the introductory analysis we're going to do, we're going to use the MAF files that we find. And a great way to do this is actually to use the CGC's data browser. And this is a graphical query engine. So on this canvas, we have 3 entities. So we start with the investigation. We narrow down to pancreatic adenocarcinoma. But of course, we could, you could search across multiple investigations if you're doing a pan-cancer study. We want to find all the cases. And then we want to find files that are open, as well as the data type is the masked somatic mutation. And the result on the bottom left is 4 files, which represent the 4 different variant callers that the Genomic Data Commons used to create these results for us. So we can then copy those files to the project and start to do our analysis. As I mentioned before, a lot of users have data that's already in the cloud. So a lot of strategies are combining your own data with some of the public data sets as well. So we see that a lot. A great way to do this, if your data is already on Amazon or Google, you could use a feature called volumes. And what this is the account on AWS or GCP. You can actually set up a certain type of user management to give read and write permissions to the platform, and then have those files appear as if they're natively in your project. But what's actually happening is it's a linking back to the original cloud storage, and the data never actually gets moved until the last moment when you do run some analysis on that particular file. So there's another way to link data, which I want to mention. And we worked with some partners at the PDC, ICDC, and CDS. And this is enabling greater multi-omic research. And you can go to the portals on these respective data nodes, and you can build your cohort there. You can do all the filtering that you need there and download a file manifest. At that point, you log back into the CGC, and you can import the files from the manifest in your Files tab inside your project. And what happens is it actually copies links to the files, and they appear as if they're in your project, but really they're linking back to the original spot in the CRDC. And so it happens very fast. And you can analyze those in a task or in a data cruncher notebook. So we talked about making a project and finding and linking your data. So let's talk about finding software to analyze your data. And there's a lot of options in this category as well. The easiest way to do things is to look in the public apps gallery. So if you go to cancergenomicscloud.org, if you look under resources, you actually will have the link to the public apps gallery, which is completely open, and you don't even need to log in to see the public apps gallery. There's approximately 500 bioinformatics tools and workflows that have been optimized for speed and cost in the cloud. And they're actually organized in different categories. So differential expression, HLA typing, metagenomics, etc. And when you click on any of the cards that I'm showing you here, you'll get a description of what the tool can be used for and what other tools it works with. In addition, you have a benchmarking section, which is generally a table that'll give you an idea for it'll give you an idea for how much time and also how much funding it'll cost to run this tool for a variety of different either file sizes or sample sizes. So that's very, very helpful. But that's not the only option. You can actually bring your own pipelines to the platform using our software development kit, part of which we call Web Composer, this is a screenshot of Web Composer here. And it's based on some things you may have heard of. So it's based on portable software environments called Docker containers. If you're doing analysis on BioWolf, you have a similar technology called Singularity. And these are portable environments that contain all the software dependencies to run a specific tool. If you couple that with a common workflow language, you can actually build out entire analysis pipelines that are dynamic depending on parameters that the user specifies, and you can pipe the results from one tool to the next. And you can do that either by writing the common workflow language yourself, but you don't have to. You could actually use the graphical canvas to do that as well. So today, we're doing a very basic math tools pipeline, and this is the graphical representation of it. So this is called a directed acyclic graph of our simplified workflow, which is a really great introduction to somatic mutation. So we're going to start with one or more math files. So when you repeat this analysis, you could choose a different investigation. It will take any number of math files. It'll read them in and create the R objects and also create a summary plot. And the results then go into one of two subsequent tools, the first being the OncoPlot and another tool being somatic signature script, which generates many outputs for us to look at, such as signature heat map, as well as detecting any ApoBec signature as well. This particular tool that I customized is not in the public apps gallery. It's on DocStore, which I had mentioned. DocStore is another great place to find workflows. So it has a lot of tools that are also in the common workflow language, and I'll show you how to do that. But we recommend any of our researchers, once you're done customizing your workflow, or if you wrapped a new tool, we recommend go ahead and sharing it with the greater community via DocStore. Okay, so I've gone over all these different categories. So now it's time to create and run your analysis. And like I said, there's two ways to do that. You could run a task, which is a completely graphical way to do it, but you can also create notebooks in RStudio or Jupyter Lab as well. So let's go ahead and switch to a live demo. I'm going to share my web browser. Everything's done in the web browser, but hopefully I've alleviated some of your concerns. So let me go ahead and share my screen. Okay. So here is the dashboard. I've already logged into the CGC, and you see I belong to many different projects, but I want to show you how quickly we can do this analysis. So we're going to make a project, and I'm going to call it MAF Demo. And then I'm going to select my billing group. Everyone should pick their pilot funds when you're just getting started, but I can choose I can choose the big customer support for today for mine. Remember, we operate on multiple clouds, so you can choose AWS or Google. The GDC actually has data on both, so that's I'll just use the default, which is AWS. And I'm going to uncheck controlled data because we're going to be using open TCGA data today. So as soon as I create a project, it takes me to this new screen, and I can invite new members. So I'll invite my colleague Sai, and I will, oops, I forgot your username. There you are. And I can fine tune her permissions if I want to. Okay, and then I'm simply going to follow the dashboard at the top. So the menu goes Files, Apps, and Tasks. So let's add some files. So files are the basis of every analysis on this platform. And I can actually add files by clicking Add Files. And this brings up the screenshot I showed you before. And we have all these different options. So we have the data browser. We have the public files, which we can link. You can clone from other projects. You can upload, directly from your computer if you have small files. If you need the data tools, you can click here, and you can attach volumes from AWS or Google here, as well as import a manifest from the PDC, CDS, and integrated K9 data comments. But today we're going to focus on the data browser. So if you see, the data browser actually allows us to select and start querying multiple data sets. So we have the latest TCGA, we have Target, CPTAC, TCIA, as well as Legacy TCGA and CCLE. So we're just going to focus today on the TCGA HG38 data. And at this point, we can start exploring. So I can type in any type of, basically any type of entity or parameter I want, or we can start with the classic, all the cases, all 11,000 cases, or any number of samples or files. You can also save your queries, which I've done before. So I have queries for pancreatic cancer as well as kidney. So I'm going to go, I already have this saved, so I'm just going to click on that. And you see that we have these 3 entities, and each entity has a variety of parameters that we can filter by. So the investigation, I've already filtered to pancreatic cancer, which automatically filtered from the 11,000 cases down to 183. And then I'm finding files that are open access and are of type masked somatic mutation. But you can go ahead and find other open data as well if you wanted to. There's, I believe, some open copy number data that you can use. And of course, if you have the correct dbGaP permissions, you can then get any level of the TCGA data. If you notice there's a unlocked icon, that means that these are open data. So anyone that logs in can access these. And if you notice, there's 4. So they're aggregated somatic information, but they're for each of the variant callers that were used by the GDC. So then I copy these files to the MAF demo project. And once again, it's actually just linking these files. It's not moving them out of the GDC just yet. So I'm going to go back to my demo project and hit refresh, and I see they're in my Files tab. And I see that I have some helpful metadata as well. And I can go ahead and if I had lots of files, I could sort by file type. So now let's talk about adding apps. So you can add an app directly from the public apps gallery, but we're going to create one really fast. And I'm going to create a workflow. So I'm going to call this somatic analysis. And it starts with a blank canvas, and I can add tools from a previous workflow or workshop, and I can add them from the public apps gallery, or I could type in the CWL code myself, or I could go ahead and paste it from another place. So I found in DocStore, I've submitted this workflow called somatic mutation analysis. So I'm going to go ahead and find the CWL file I want to use, and I paste it here, and I automatically get the visual editor. So these represent different R functions, somatic signatures, the oncoplot, and reading in the MAF file. So I'll go ahead and save this and give it a run. So we're just about done here. So I'm going to select my MAF files, and I can choose, and this will demonstrate how you could actually run a tool across multiple files. So we have 4 MAF files here. I'm going to turn on batching, which is going to automatically create one task for each of these files. So later on, we could compare, let's say, the different variant callers and to see which gave us the better results. So I click run, and it automatically creates 4 child tasks. Okay, and those these tasks run very quickly. I actually executed them in another project, and I just want to show you the results. So I was able to analyze all 4 of them very, very quickly. Let me find Mutech. So with Mutech, it ran in 5 minutes, and it cost 3 cents of the pilot funding. And then we have all these outputs. So the outputs are viewable also directly on the platform. So you don't need to download anything. So I can look at my oncoplot. So I can open that in a new tab. And I can see the usual suspects of KRAS, TP53, SMAD, SMAD4, and CDKN2A. And I can see that as well. And then we could explore the somatic signature using, which one should I pick? This one. So this is a really great, MAFTools is really, really great. Provides a lot of information for you. So this one is showing that we get C to T, somatic changes, which is related to enzymatic deamination of 5-methocytosine, which is correlates to usually the age of the subject. So at this point, I'm going to switch back to slides and just finish up showing you the oncoplot again, and mentioning that you can actually do this analysis using RStudio or JupyterLab. And the way that works is you actually can spin up a private instance of either one of those softwares, and you can access all the files in your projects as if they were natively available to you. So we did a lot of things today. We found TCGA open data, we imported a tool, and we ran a pipeline. And really, there's a lot of stuff that we've demonstrated, including connecting multiple tools together. And I just want to mention before we take some questions that we're starting up a new office hours. So office hours will take place every week. So the next session is tomorrow at 3 p.m. Eastern time. So Thursday, January 28th. And you can find the link at cancergenomicscloud.org/office hours. We'll tell you how to log in. So we can do, please bring questions, intro questions, or more advanced questions. It's up to you. We can do some more TCGA open analysis, such as copy number variation, etc.
Okay. Thanks so much, Dave. That was an excellent overview of the CGC features, and I loved seeing the demo as well. I think this was the last thing I wanted to tell you, so thanks for staying till the end. So I was telling you, if you wanted to learn more about the PDXNet And if you're excited to hear the PDXNet stories, so please join us for the next month's webinar, which is going to happen on the 24th. Dr. Dennis Dean, who's a principal investigator at 7 Bridges, will be talking about how PDXNet helps with advancing preclinical trials on the Cancer Genomics Cloud. So with that, we are going to answer some of the questions that have come up in the chat. So let me start with, okay, so are any of the publications using CRDC data? So I mentioned that during my presentation. There are several users that have used data from the genomics data commons or the proteomics data commons. And Marcia, I see your follow-up question. I'll certainly send the references to you. There's another question about why don't I see Genie or All of Us or N3C? So this is a great question. So we've certainly considered using Genie. So bringing Genie and integrating it, it's available as part of the genomic data commons, but there are some policies that we are reviewing. So we'll consider and explore that. With respect to All of Us, we're very interested in bringing that, and we are participating in the NIH interoperability efforts. So we want to see how we can connect with an entirely different ecosystem where, All of Us is in a different ecosystem. So we're certainly looking into that as well. N3C is sort of relatively new, and we have started looking into N3C as well. Although, there's a lot of COVID data, we have to see how it aligns with the CRDC efforts. Okay, and I'm also going to take this other question. Is the CGC certified for PHI? Dave, do you want to take this?
So the short answer is it depends. And I think it depending on how you use a system, it absolutely can have the patient data uploaded, but it's probably better to have a separate call to go over the specifics.
Yeah, and also, there are policies outlined in the CGC website, so I would highly recommend you to look at those, and we can certainly discuss that. Will you provide the recording of this webinar later? Yes, absolutely. We will post it in the same place, cancergenomicscloud.org/webinars. So we'll post the recording and the slide deck. How does the CGC differ from Cavatica? They look similar. Dave, do you want to take this?
That's a great question. So we offer a variety of academic platforms that operate to different communities. So Cavatica is another platform that serves another community, primarily focused on pediatric rare disease, and Gabriella Miller Kids First data. We also have a similar looking platform called the Biodata Catalyst, powered by 7 Bridges, which is based around the community for the top med data sets. So, and that's sponsored by the NHLBI. So we have a variety of platforms depending on your research interest, and some users actually span across platforms. So if they want to hook up to different data sets.
Okay, so we're almost at time, but I just want to get to this one last question. What are the differences between CWL and WDL?
Great question. So they essentially do the same thing. So they're both workflow specification languages, and they're both available on DocStore. So the 7 Bridges platforms run common workflow language, but some other platforms run the workflow description language.
All right, great. Thank you so much. And thanks everybody for joining and staying with us. We look forward to seeing you again in the next month's webinar. Bye.